So before I dive into Sequenomics, I want to just give a little bit of background on Watchmaker. So we are a genomics tools company headquartered in Boulder, Colorado, with over 170 employees worldwide. We are focused on improving reagent quality for high stringency molecular biology applications where data quality is paramount and there is a large scope for improvement over existing solutions. Now, ideally, your data output would be a perfect representation of the information contained within your biological sample, but that's inherently challenging. The reality is that all biological information is read, written, and maintained by enzymes, and to access that information requires multiple enzymatic manipulations. These steps can be lossy and can result in data corruption. Now, we believe that if you can improve the quality of the enzymes, you can improve the quality of the data. And so enzymes are at the core of Watchmaker's precision products development platform, wherein we are leveraging our expertise in computational biology, enzyme engineering, and enzyme manufacturing to purpose-build enzymes for specific applications whose workflows are developed in parallel. And we use the needs of those applications to define the performance requirements of those enzymes. Now, this is a complex engineering effort, and it is very challenging, but it has a lot of benefits. Vertically integrating our engineered enzymes into highly optimized end-to-end NGS workflows confers performance benefits, specifically and importantly when you're working with challenging inputs and samples like low input material and FFPE. But it also has the benefit of improving our costs and our supply chain consistency. So because we're not sourcing polished, finished goods like enzymes from other suppliers, we're able to control and keep our costs low, have better control over our supply chain, and we can pass on those benefits to our customers. Now, beyond the cost of library preparation kits themselves, there are a number of factors that play a role in NGS Sequenomics. One important one are operational costs and considerations. Things like turnaround time, personnel and instrumentation occupation times, the ability to automate and scale easily to kind of, realize economies of scale. Additionally, reducing downtime as a function of back orders due to supply chain inconsistency or performance issues that require troubleshooting. Inventory management is another piece, needing to carry and manage inventory that spans multiple library prep chemistries. Being able to simplify and streamline that inventory management has a lot of benefits. And even ease of implementation, right? When you're looking to set up a new workflow in your lab, there are startup costs associated with that. And so choosing a solution that is easy to kind of get off the ground can reduce your startup costs overall. But the reality is that these cost benefits or reducing costs can't come at the expense of sequencing quality. Data quality is still paramount. And so what we see is that these concepts of workflow, ease, performance, data quality, and price are often competing. You can maybe get 2 of these in a single chemistry, very rarely 3, and that's something that Watchmaker is working to change. So first we'll start with DNA Sequenomics. Now, one of the most impactful things labs can do to reduce their sequence costs overall is to make the switch from a sonication-based or Covaris-based DNA fragmentation method to enzymatic fragmentation. This omits any instrumentation costs and the associated maintenance costs. It omits any specialized consumable costs, and it also alleviates a bottleneck in your DNA library preparation workflow, so it can simplify your processing as well. Now, I will say that enzymatic fragmentation has a bit of a bad reputation. A lot of traditional solutions, while they provide that scalability and ease of workflow, they come at the expense of sequence accuracy, which nobody wants. And then also they can prove challenging to implement across a wide range of DNA inputs, in terms of mass and quality. And so we have worked hard to develop the Watchmaker DNA library prep kit with fragmentation that harnesses those scalability benefits of an enzymatic fragmentation workflow, but doesn't sacrifice sequence accuracy or ease of processing across variable DNA samples. So tuning your library size is as simple as modulating your fragmentation time and temperature, which we're showing on the left. And this eases operations, it eases your startup time. On the right-hand side, what we're showing is the robustness of this chemistry across a wide range of DNA input amounts. So we're looking at 100 picograms up to 500 nanograms using the same fragmentation conditions and showing very equivalent library sizing. And so this means that you can implement this workflow and it's robust and will perform well across a very wide DNA input range. It just simplifies your overall processing, enables sample batching, all that good stuff. As I mentioned, sequence artifacts or sequence accuracy tends to suffer with enzymatic fragmentation solutions. One of the types of artifacts that can form are chimeric reads. So these are reads that map to multiple locations in the genome. And when they're real, they can indicate structural variants or translocation events. But if they're really just a function of the library prep workflow that you're choosing, they're noise in your data. On the left-hand side here, we're just showing a reference from a webinar where the speaker did a comparison between the same core library prep chemistry, but just fluctuated whether or not they did Covaris shearing up front versus enzymatic fragmentation. And what they saw was almost a tenfold inflation of chimeric reads. So it's a lot of noise and, again, wasted information. And so we kept this in mind during the development of the Watchmaker kit. And what we're showing on the right is the performance of our solution in purple relative to a Covaris control, so that gold standard for sequence accuracy, and to other commercially available enzymatic fragmentation solutions. And what you can see is that we have chimeric read rates that are on par with sonication and much lower than other commercially available enzymatic fragmentation solutions. Another type of artifact that can form are something that's called hairpin artifacts. So one proposed mechanism for how these artifacts form is shown on the left. In short, essentially if you have exposed single strands on your molecules and there's any type of maybe semi-palindromic sequence, you can get self-annealing and then the end repair machinery can stitch that molecule together and you wind up with basically like an intramolecule chimera. This has implications for performance with variant calling assays in particular. So on the right-hand side is a published assessment of, again, the same core DNA library prep chemistry, varying only whether or not it's Covaris used up front, which is shown on the far left of the plot, or enzymatic fragmentation shown on the far right of the plot. And again, you can see there's a massive inflation in the number of variant calls, just as a function of your choice and how you fragment your DNA upstream. And so again, we kept this in mind during development of our kit. We developed a bioinformatics solution that specifically can detect these hairpin artifacts. And what we're showing here on the right is, again, a comparison of our enzymatic fragmentation solution relative to that gold standard Covaris control. And we can see rates that are on par with Covaris, and again, much lower than other commercially available enzymatic fragmentation solutions. So overall, we're able to harness those workflow benefits of enzymatic fragmentation and maintain sequence accuracy that you've come to expect from a sonication type workflow. The other, implication or impact of having these hairpin artifacts in your sequencing data is the prevalence of something called soft clipping. So soft clipping happens when the ends of your reads don't align to your reference, and they're bioinformatically trimmed, basically, to get to a sequence that does map very well. And all of those trimmed bases are wasted bases. It's sequencing economy that you're not utilizing. And so they can be, this mismatch, this soft clipping can be inflated as a function of just having these hairpin artifacts present in your library. So what we're showing here is soft clipping data for a set of FFPE samples. We looked at FFPE samples spanning a range of qualities, from high quality, designated as HQ, down to low quality, designated as LQ. And we're comparing the Watchmaker enzymatic fragmentation method to Covaris shearing. I think the first thing that you can take away here is that, in general, when you're looking at FFPE performance relative to a high-quality control like NA12878, you can see a general inflation in the amount of soft clipping that happens. And you can imagine that this is likely due to the amount of damage in FFPE samples that are causing a number of exposed single-stranded ends on your molecules. The other takeaway from this data is that, Covaris on its own with FFPE samples has, quite high soft clipping rates. And you're able to actually, minimize soft clipping, better maximize your sequencing economy just by implementing an enzymatic fragmentation solution. And so here what we're saying is, not only do you have the processing and the scalability benefits of enzymatic fragmentation, but if you're working with FFPE, you could actually be improving on your data quality as a function of using enzymatic fragmentation over the gold standard Covaris. I would say another consideration for improving your sequencing economy, besides reducing soft clipping, events, is, in minimizing the GC bias that's associated with your DNA library prep workflow of choice. So here we're showing a PCR-free whole genome sequencing data set that's comparing our enzymatic fragmentation solution, again in purple, to the gold standard Covaris in gray, and then a subset of other commercially available solutions. We're looking at 300 nanograms of a high-quality control input, and we also evaluated our chemistry with 75 nanograms because what we saw was very high-efficiency library prep, and we were able to get basically, a sufficient quantity to go into downstream sequencing with, a quarter of the input mass. Specifically, what we're measuring here is the mean coverage of challenging promoters. So this is a published set of GC-rich promoters that are typically undercovered in traditional sequencing data sets. And so, I think the first takeaway is that the Watchmaker chemistry does a great job of covering these challenging regions. All of these libraries saw the same number of sequencing reads, but Watchmaker is giving you improved coverage of these GC-rich areas. And this translates to sequencing economy. You can imagine if you need a minimum coverage depth of 30x across your genome, and you're undercovering these GC-rich promoters, you need to sequence more to get that minimum coverage depth that you're looking for. And so this directly translates to sequencing costs at the end of the day. Now, these coverage benefits also extend to our Watchmaker DNA library prep kit. This does not include enzymatic fragmentation. This workflow is designed to work with pre-fragmented samples like cell-free DNA, or to be used downstream of sonication. Here we're looking at a 25 nanogram mock cfDNA sample. And what you can see on the left is for equivalent sequencing reads, overall improved coverage with the Watchmaker solution relative to other commercially available solutions. And this directly translates to the right in improved coverage of rare variants. So these variants are present in this sample at a 0.1% allele frequency, and so these are very low frequency variants, and you can see a substantial improvement in the coverage depth of those variants with the Watchmaker chemistry. Now, this is great because it actually gives you flexibility in how you choose to sequence. So if you're sequencing currently and you make the switch to Watchmaker and you get improved coverage for the same number of sequencing reads, you have two choices. You can either sequence less to get your desired coverage depth, so you're saving on sequencing costs, or you can continue to sequence to that same number of reads, but actually get improved coverage of maybe rare variants that you're looking for. So it gives you the flexibility that you need and want in a sequencing workflow. So I'm going to take a break and take a sip of water. Moving on to RNA sequenomics. I think one of the biggest areas of opportunity in RNA sequencing when we're talking about reducing overall costs is actually in improving the workflow. So traditional RNA sequencing workflows are labor intensive, they can take multiple days to execute, and that obviously carries operational costs associated with it. So here we're showing the workflow for our Watchmaker RNA library prep kit with Polaris depletion. This is a whole transcriptome sequencing library prep solution where we're depleting ribosomal RNA and globin mRNAs. And we're comparing our solution in the top row to other commercially available solutions down below. And I think the first takeaway is just the overall improvement in turnaround time. So depending on who you're comparing to, we're able to save you up to 3 hours of overall turnaround time, which is great. That means you can kind of get sample prep truly done in a single day with enough room for setup in the morning and maybe QC in the afternoon. The other, I think, impact of this turnaround time improvement is on, again, personnel and instrumentation occupation times. So your people and your instruments can be doing other things than RNA library prep as a function of how fast this workflow is. It also has, overall, a reduced number of handling steps. and a reduced number of cleanup steps. And so this translates directly to reduced consumable use. So per 96 libraries, we can save you about 1,000 pipette tips. And we also reduce the amount of beads that you need because we have fewer bead cleanups throughout the protocol. This simplified workflow also translates to ease of automatability. The fact that we have fewer handling steps means fewer user errors and needing to rework samples, let's say. And also, if you think about maybe onboarding in your lab, it's much easier to train and onboard somebody on a fast, simple chemistry than it is, a very onerous, time-intensive, and multi-day workflow. The last thing I'll mention here is that this chemistry is compatible with RNA inputs ranging from 1 nanogram up to 1 microgram, and it's also been optimized, and we have a specific protocol for working with FFPE material. And so this kind of gets back to that idea of simplifying your inventory management, right? Now you can take one kit and address a majority of your RNA sequencing needs, whereas historically you may have needed niche workflows or niche chemistries to kind of hit all of your sample requirements and application requirements. What's really nice is that this workflow improvement and streamlining doesn't come at the expense of data quality, and in fact plays a role in enhancing data quality. So here we're looking at a whole blood derived RNA sample with inputs ranging from 1 nanogram up to 500 nanograms. And on the left, we're looking at wasted bases. And so we're classifying wasted bases as any base that passes filter but doesn't align, or maps to ribosomal RNA or globin mRNA. So these are bases that are not useful for you. And the first thing that you'll see or notice is that as you drop your input amount, the amount of wasted bases, either due to depletion inefficiencies or maybe the creation of just junk, truly starts to inflate as you get down to maybe that 10 nanogram, 1 nanogram input amount. And that's not true for the Watchmaker chemistry. You'll see a slight inflation because no chemistry is perfect, but we don't really fall off a cliff, and we're very efficient overall in terms of maximizing your sequencing economy. And so this also translates to improvements in sensitivity. So on the right, we're looking at the number of unique genes detected with all of these chemistries. And I think that the main takeaway is that Watchmaker performs very well across this full range, and in fact outperforms when you're talking about moderate and low input amounts. And these performance improvements extend to FFPE. So here we're looking at a matched fresh frozen FFPE sample set. And we're looking at libraries prepared from 10 nanogram inputs. On the left, we're looking at unique genes, and the takeaway here is Watchmaker detects more unique genes across both sample types with this very limited input mass. But what's really interesting is when you start to compare those FFPE and fresh frozen libraries. So you can use those fresh frozen libraries as a sort of control. They tell you what should be in your FFPE libraries that you may be missing. So when we do this overlap analysis, which we're showing on the right, what we find is that in the Watchmaker FFPE libraries, we're able to detect 73% of the genes identified in the fresh frozen control material. And that's a much higher percentage than what's detectable with the other chemistries that we've assessed. So you're really able to maximize your insights and maximize the information out from this very challenging input, both in terms of quality of RNA and mass, and again, maximize really what you're getting out of your sequencing. So there's a similar story when we look at our mRNA library prep workflow in terms of workflow simplicity and in terms of data quality. Again, we're showing a snapshot of the workflow here, and one of the first takeaways is, again, the reduced turnaround time. So we can save you up to 3 hours of turnaround time depending on the workflows that you're comparing to. And again, that comes along with personnel and instrumentation occupation times and reduced consumables use. Again, much like our whole transcriptome solution, this kit is broadly compatible with inputs ranging from 2.5 nanograms up to 1 microgram. And so again, you can likely simplify your inventory management and carry one chemistry that can do a lot rather than multiple chemistries that do very unique things. And again, this streamlining doesn't come at the expense of data quality. So here we're looking at a breast tissue-derived RNA sample from inputs ranging from 2.5 nanograms up to 100 nanograms, looking at both duplication and unique gene detection. And so, on the left-hand side, looking at duplication, the first thing that we can take away is that the Watchmaker chemistry significantly reduces the amount of duplication in the libraries, and that helps to maximize your sequencing economy. And it also translates to improvements in sensitivity, which you can see to the right. Across the board, we're able to detect more unique genes. And then I think what's also really interesting, and this applies to both chemistries, I only wanted to show it for one, is the fact that the Watchmaker library prep solutions are actually able to maintain quantitative gene expression data as you drop your input masses lower. So these are differential expression MA plots. Here, our test condition are 10 nanogram libraries. Our control condition is 100 nanogram libraries. And we're looking at the differential expression between those two sets of conditions. And just to orient folks who aren't familiar with these MA plots, on the x-axis is a measurement of gene abundance. So every dot on this plot is a gene. The x-axis is a measurement of abundance. So on the right-hand side are higher abundance genes. On the left-hand side are lower abundance genes. Along the y-axis is a measure of the fold change of that 10 nanogram library relative to 100 nanograms. So that log fold change zero line means there's equivalency, and anything above that log fold change zero line is more abundant in the 10 nanogram sample relative to 100 nanograms. Anything under that line is less abundant in the 10 nanogram sample relative to the 100 nanogram sample. The color coding here, genes that are color-coded in a darker shade are actually being picked up as differentially expressed in the 10 nanogram samples. And so in an ideal situation, what you would see is all of your genes collapsing on that log fold change zero line, meaning you're getting the exact same information out at 10 nanograms as you are at 100 nanograms. And nothing is ideal, but the Watchmaker chemistry here does perform very well. So what we see is a normalized distribution of these genes around that log fold change zero line with no systemic bias or upswing or downswing in the data. And then very few genes being picked up as actually being differentially expressed. In the other chemistries that we've assessed, what we actually see is a bit of an arm that forms. Let's see if I can point it out. Yeah, an arm that forms here, especially for lower abundance genes. And what this is saying is that you're actually losing relative coverage of these lower abundance genes in the 10 nanogram libraries relative to your 100 nanogram libraries. And there's a much higher number of genes being picked up as being differentially expressed. And so the takeaway here is that you can prepare libraries from 10 nanograms of RNA with all of these chemistries and sequence them, but it's really only the Watchmaker chemistry that's maintaining that quantitative gene expression information that aligns more closely with higher inputs and then ideally more with that original biological sample. Another thing that we do at Watchmaker that I think is maybe a bit unique is we really consider scalability and we bake it into all of our products and really kind of how we operate as an organization. And so I've already covered the workflow piece of things where we have simplified workflows and that eases handling, it reduces consumables, all of those great things. One of the other impacts of having these very streamlined workflows is that even when you're processing a 96-well plate of libraries, you can do so in under a day, even with our longest NGS protocol at this point. Beyond the workflow itself, we pay close attention to how we design our protocols in development to make sure that they're compatible with major liquid handling platforms. So specifically things like having minimum aspiration and dispense volumes. Make sure that you can robustly execute this across a variety of robots reproducibly. We're not asking you to pipette 1 microliter, let's say. In terms of our kit configurations, our higher throughput kits, like the 96 reaction kits, are designed with a generous overage, usually around 20%, and this is to help account for dead volume requirements across different liquid handlers, as well as being able to have multi-use use of these kits. We also do very rigorous reagent stability testing, to ensure on-deck performance, and that includes both the individual components as well as master mix stability, to just really ensure that they're highly automatable. Now, beyond the way that we design our products for scaling, we've also structured a team specifically dedicated to automation support. And so this team, does a whole host of things within our organization. One is that they provide consultative support to customers. So, if you're looking to build your own automated workflow. They can answer questions, they can provide tips and tricks, and really kind of help you on that path. They can also provide liquid class testing information, both at specific values and recommendations for aspiration, dispense, and mixing. And then I think one of their biggest impacts is really their ability to install and train on all of our standardized methods. So this includes both remote and on-site installs. And these methods are always developed in partnership with these third-party liquid handling platforms, and so you're ensured cohesive support from both Watchmaker and the liquid handling organization. Lastly, a big focus for them is in actually helping to develop those protocols. So they spend a lot of time partnering with these automation organizations and building out our library of automated methods to ensure that we have the solution that you need for the platform that you're working on. So this table is set up looking by chemistry as well as by the specific platform. And the way that you would read it is anything that has a check mark has been developed, vetted, and has been successfully installed in the field. So we don't consider anything finished until we've actually installed it at a customer site with success. Anything with an R is a method that's ready to go with everything except for the fact that we have not implemented it externally just yet. So if you're interested in any of our programs, reach out to us. And then anything with a D is in development. So we're still working to build the methods, test, and optimize those methods. And this is an ever-expanding list. We actually update our website monthly, I would say, with new methods that are available. So it's a really active program that we have. And this, I think, really ties a lot of these themes together. So here we're looking at, again, the Watchmaker RNA library prep kit with Polaris depletion. Up top is that manual workflow that I showed previously. The time is estimated based on 8 libraries being prepared at once. And then down below is the turnaround time for 3 independent automated liquid handling methods across 2 different platforms. And so I think the biggest takeaway for me is that each of these platforms has a different deck layout, different design, different capabilities, but we're able to process a full plate of 96 whole transcriptome libraries in under 7.5 hours. And so that gives you the time to do the setup that you need to do in the morning and really makes this executable in a single day. Okay, so now I'm going to change gears a little bit and just talk about some of the areas of continued innovation at Watchmaker. So along the same thread of enabling and supporting high throughput operations, we are currently developing a normalization solution. And I'll just walk through a bit of the mechanics of it here. It's very streamlined. It involves the binding of a normalization reagent and then a subsequent bead capture of that normalization reagent, some washing, and then elution. And this results in the equimolar normalization of up to 96 libraries in as little as 1 hour with a highly automatable workflow. Now, some of the features here are kind of the things that it doesn't require. So it doesn't require additional PCR. It doesn't require additional equipment. You don't have to do any variable volume pipetting. You don't need to waste your time on dilution and pooling calculations. And it's a non-destructive method, so you're not destroying the library and you're not single-stranding your library. So I think it's a really novel approach to library normalization. On the left-hand side, we're just showing some data coming out of our development team. So we're looking across a 20-fold library input into normalization range. I believe these are actually RNA-seq libraries. And so what you can see is for that 20-fold input range, we're able to collapse the CV to 14% in terms of your sequencing read distribution. I think we've actually had some data sets since then that are actually much more tight than 14%. But it's, I think it's it's a great chemistry, and we're really excited to be commercializing it later this year. Our other area of focus is in the realm of methylation sequencing, specifically in the development of our TAPS chemistry. And so those not familiar, TAPS is unique, and I would say kind of flips the script on methylation sequencing relative to bisulfite or EM-seq, chemistries, in that those traditional chemistries convert all of your unmethylated cytosines and protect your methylated cytosines. And our chemistry is actually the opposite of that. So instead, we are protecting your unmethylated cytosines, and we're directly converting those that are methylated. We've made a lot of improvements to this chemistry over the published method. And just to outline a few, one is swapping in our Watchmaker DNA library prep kit to improve the conversion of DNA into sequenceable library. We've also undertaken some TET engineering efforts to improve activity as well as stability of that enzyme. We have swapped in a novel borane that is much less toxic than the borane that was used in the original publication, and this lends itself to being used in just standard lab settings. And then lastly, we're using a highly optimized and engineered library amplification solution that can handle amplifying DHUs, which are pretty difficult to amplify through. And all of these things kind of culminate in a very streamlined single-day workflow where you can produce sequencing-ready libraries in under 7.5 hours, and again with a very automatable workflow. Because it's less damaging to your template than something like bisulfite sequencing is, you're able to work with inputs as low as 1 nanogram, and that opens the door to methylation profiling from samples like cell-free DNA or FFPE. And then lastly, again, it's a direct conversion of those methylated cytosines, and we have a very high conversion efficiency. So just a little bit of background. CPG islands, which is where, the majority of methylated cytosines reside, account for about 20,000 genomic regions. And that translates to about 0.8% of the human genome. On top of being, fairly rare, they're also non-evenly distributed across the genome. So you can see, over 50% of the genome has no CPGs in a 100 base pair window. So what that means is that when you're working with a chemistry like bisulfite sequencing or EM-seq, and you're converting all of your unmethylated cytosines, you're actually converting 22% to 23% of your bases, and you're collapsing a 4-base genome into a 3-base genome. Conversely, with TAPS, where you're actually converting those methylated cytosines, you're only converting 1% to 2% of your bases, and you're able to maintain that 4-base genome complexity. And so this is really the part of TAPS that helps to unlock this idea of multimodal analysis, where you might be able to access variant calling type information in addition to methylation status information from a single library. And this, I think, illustrates that point really well. So down below, for the same genomic region, you can see bisulfite sequencing and EM-seq with a number of Ts being called as base substitutions. And up top with TAPS, again, you can see that it's maintaining that 4-base genomic diversity, and you can actually start to visually resolve where some of those variants might lie. So this is looking at data across both hypo and hypermethylated human genomic regions, including those covered by the EPIC array, as well as just CPG islands in general. And the takeaway here is when we're looking at the TAPS data relative to bisulfite sequencing and EM-seq, we see the ability to detect both hypo and hypermethylation, and we see very comparable performance in terms of the data output to these traditional methylation sequencing approaches. And then lastly, so we wanted to really probe the possibility of using this chemistry for variant calling. And so we basically executed a comparison study between a tumor normal colon tissue sample set. We looked at our control workflow, which is just our standard DNA library prep kit, and then compared it to a TAPS workflow. And the first thing that I would take away is that we are able to pick up variants with the TAPS chemistry. And I think really notably, these first 3 variants are actually C to T base substitutions, which would be very, very challenging, if not impossible, to pick up with traditional methylation sequencing. I think the other takeaway is the fact that these are very low allele frequency variants, and there's actually really great concordance between the TAPS chemistry and the control chemistry. And so it really does provide proof of concept that you could potentially access somatic variant calling with the TAPS chemistry, in addition to methylation profiling. And so just quickly to summarize, this chemistry enables multimodal analysis. It reduces damage of DNA, opening the door to cell-free DNA and FFPE. It improves your sequencing economy because you're not losing as many bases or as many reads to being low complexity. And it's a single-day workflow, so highly streamlined, highly automatable. It really kind of holds to all of Watchmaker's tenets in terms of product development.